In this lecture, we are finishing up Chapter 18 on the electron transport chain by focusing on sections 3 and 4 regarding oxidative phosphorylation and the regulation of the electron transport chain. As you may recall from the previous lecture, the electron carriers NADH and FADH2 are oxidized to ultimately transfer their electrons to oxygen, making water. In this video, we are focusing on how this flow of electrons is then coupled to oxidative phosphorylation utilizing the enzyme ATP synthase, and then how this whole process is regulated by the cell. After completing this unit, you should be able to meet these listed learning objectives. The activities we do in class will then build upon these learning objectives in order to allow you to begin analyzing and discussing the relationships between electron flow and ATP production in the mitochondria. Distinguish between coupled and uncoupled mitochondria, interpret isotopically labeled carbon dioxide data from peer-reviewed literature, and to further develop and practice problem-solving strategies utilizing your peers' diverse backgrounds in order to solve interdisciplinary problems. As you recall from the previous lecture, electrons are cascaded through the electron transport chain through a series of redox centers, eventually reducing oxygen to make water. At the same time, as the electrons are flowing through the electron transport chain, protons are being pumped against the proton gradient from the mitochondrial matrix into the intermembrane space of the mitochondria. This process is endergonic and occurs by coupling this process with the incremental energy that is released as electrons flow down the electron transport chain. There is only one route by which the protons can flow back along the gradient into the matrix, and that is through the enzyme ATP synthase, or ATPase. The free energy that is released during this process is harnessed by ATP synthase to create ATP from ADP in inorganic phosphate. This process I just described is called the chemiosmotic theory that was first proposed by Peter Mitchell in 1961, where it was met with strong resistance by other scientists in the field. Mitchell's theory was the first to propose a phosphorylating process that was governed by a membrane-bound enzyme powered by a proton motive force and not a free-floating soluble enzyme. It took nearly 20 years for Mitchell himself and other scientists who believed in him to provide concrete evidence of his theory, and finally in 1978, he was awarded the Nobel Prize in Chemistry. There are four key principles underlying the chemiosmotic theory. 1. You must have an intact inner mitochondrial membrane. Any hole or holes in the membrane would allow protons to freely diffuse back into the matrix as they are pumped, disrupting the proton gradient. The membrane must also be impermeable to other charged ions such that they cannot freely diffuse and disrupt the charge gradient created by pumping the protons. The process must be coupled with the electron transport chain, which is the mechanism that pumps the protons from the low concentration matrix to the high concentration intermembrane space. 4. Compounds that increase the permeability of the inner mitochondrial membrane act to uncouple the effects of the electron transport chain by allowing protons to freely diffuse back into the matrix and thus inhibit ATP synthesis. In this instance, the electron transport chain will continue to function, but ATP synthesis will stop. This is what happened in our case study we reviewed in our recent class where patients were poisoned by the molecule DNP. DNP acts as a decoupler by binding and carrying protons through the membrane. As you recall, DNP causes a hypermetabolic state by uncoupling oxidative phosphorylation. Energy is released in the mitochondria as heat, and toxic doses of DNP will result in uncontrolled thermogenesis, leading to hypothermia and systemic responses to elevated body temperature. The proton gradient that is created during the electron transport chain is an endergonic process. However, the flow of protons back across the membrane through ATP synthase releases energy that can be used to make ATP. The phosphorylation of ADP to ATP requires approximately 40 to 50 kilojoules per mole. This amount of energy requires the energy released of at least 2 protons re-entering the matrix per molecule. The enzyme complex that governs this process is called ATP synthase. It is a multi-subunit transmembrane protein that is around 450 kilodaltons in size. The transmembrane portion of the protein that is lipophilic is called the FO and consists of 8 subunits. The water-soluble portion of the protein that extends into the matrix cytoplasm is called the F1 region and consists of 5 subunits. A number of biochemists liken the structure of ATP synthase to a lollipop. Looking closer at the F1 portion of ATP synthase, the portion that extends into the cytoplasm, we can see that it is comprised of 3 alpha, 3 beta, 1 gamma, 1 delta, and 1 epsilon subunit. The alpha and beta subunits are nearly identical to each other and are alternately arranged together like segments of an orange around the upper portion of the gamma subunit. The arrangement of the alpha and beta subunits gives rise to a pseudo-threefold symmetry, although this protein is not actually symmetrical because the alpha-beta dimers each adopt different conformations with different substrate affinities that we will analyze later on. The transmembrane F0 subunit is comprised of 1A, 2B, and 9 to 12 C subunits. The C subunits associate to form a ring that is embedded in the membrane. The epsilon subunit of F1 interfaces between the C ring of F0 and the gamma stem of F1 to create a foot-like interface connecting the 2 portions of ATP synthase. The 2 portions are also connected via the delta portion that interfaces with the alpha-beta dimers of F1 and with the B helices that are anchored to the highly hydrophobic A subunit. ATP is synthesized by the coordinated interactions between F0 and F1 components of ATP synthase. Translocation of the protons is carried out by the F0 subunit, while the formation of the phosphoanhydride bond of ATP is catalyzed by F1. The catalysis to make ATP from ADP is governed by a binding change mechanism. In this mechanism, each of the alpha-beta subunits alternate between one of 3 conformational states. The L state is where the substrates ADP and inorganic phosphate enter and are bound loosely. The T state is where the substrates are brought closer together, are bound tightly, and is when catalysis occurs to form ATP. The O state is where the product is released and new substrates will not bind. The free energy that is harnessed as protons flow through the F0 component of ATP synthase is what drives these conformational changes in the alpha-beta dimers to go from the L to the T and finally to the O state, and then the cycle starts over. The reaction forming ATP is essentially at equilibrium under the conditions of the enzyme's active site in the T state. Due to this alternative mechanism, we can theoretically catalyze the phosphorylation of a continuous stream of ADP molecules as protons flow back from the inner membrane space to the matrix through the FO subunit. The overall mechanism of ATP synthase can be likened to a rotary engine, where the rotor is the C ring in association with the gamma and epsilon subunits, and the stator is the AB2 subunits in association with delta and alpha beta dimers. In order to drive the rotation of this engine, protons from the inner membrane space enter through the A subunit through a hydrophobic channel and bind to one of the C ring subunits. This binding causes the C ring to rotate incrementally. As more protons bind, the C ring continues to rotate until it reaches a second hydrophobic channel in the A subunit that opens to the matrix, and the proton then disassociates from the C ring through the A channel into the matrix. The C ring then continues to turn as more protons enter from the inner membrane space. As the C ring rotates, it drives the conformational changes of the alpha-beta dimers through structural associations with epsilon and gamma. If ATP synthase generates 3 molecules of ATP per full turn of the C ring, and there are approximately 10 C subunits, then the passage of each single proton provides enough energy to generate approximately 0.3 molecules of ATP. Hence, this is why we do not see whole numbers of ATP synthesized for each molecule of NADH and FADH2. Rotation of the C ring against the A subunit is driven by the interactions of an aspartic acid residue on the C subunit and an arginine residue on the A subunit. In the absence of a proton, these two residues form electrostatic interactions that bring the C subunit towards A. However, as the proton enters into the C subunit, it binds to that aspartic acid residue, neutralizing the electrostatic interactions and allowing the next deprotonated C subunit to be attracted to the A unit and driving the incremental rotation. At this point, we have investigated the complete metabolic breakdown of glucose. If we are able to derive 2.5 molecules of ATP from 1 molecule of NADH and 1.5 molecules of ATP from 1 molecule of FADH2 through coupled oxidative phosphorylation with the electron transport chain, how much ATP total can the complete oxidation of 1 glucose molecule produce? Assume that we do not go through the pentose phosphate pathway or any other detours from glycolysis or the citric acid cycle. Also remember, don't forget to consider the ATP that is generated from substrate level phosphorylation. As we have seen, the coupling of the electron transport chain to oxidative phosphorylation is vital to synthesizing ATP. Uncoupling agents such as DNP increase the permeability of protons, thereby disrupting the gradient and hindering ATP synthase from completing its function. Uncoupling agents can increase the permeability of protons by either creating holes in the membrane or by binding protons and freely diffusing themselves across the membrane, carrying the proton with them, such as we have seen with the molecule DNP in our case study. The majority of control of the metabolism of glucose to make ATP exists in glycolysis and the citric acid cycle. All of the steps in the electron transport chain all operate near equilibrium and are therefore not targets of inhibitors or activators. However, ATP synthase can be inhibited by the self-oligomerizing protein IF1. In actively respiring mitochondria, in which the matrix pH is high, because we have a low proton concentration, IF1 is an inactive tetramer form. But if the pH decreases below 6.5, the tetrameric protein dissociates into dimers, which can then bind and inhibit ATP synthase. This is a particularly useful mode of inhibition because if the matrix pH is falling, this is an indication that protons are accumulating in the matrix at a rate faster than they are being pumped out, which may be an indication that the electron transport chain is slowing down as NADH and FADH2 concentrations are reduced. Probably because the citric acid cycle and/or glycolysis has been inhibited due to sufficient ATP concentrations in the cell. If flux through glycolysis and the citric acid cycle increase due to changing demands of the cell, then more NADH and FADH2 will be made. The flux through the electron transport chain will increase. Protons will again be pumped out of the matrix, increasing the pH of the matrix. This increase in pH will then cause deprotonation of IF1 protein, which will allow the dimers to reform into tetramers, thus no longer being able to inhibit ATP synthase. Now might be a good time to take a step back and review how glucose metabolism is regulated in glycolysis and the citric acid cycle. In glycolysis, we can inhibit hexokinase, phosphofructokinase, and pyruvate kinase. The step between glycolysis and the citric acid cycle that is governed by pyruvate dehydrogenase can also be regulated by inhibitors and/or activators. In the citric acid cycle, we can inhibit citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. By utilizing these points of control, we can also regulate the electron transport chain as well as ATP synthase by default. Over our next 2 class meetings, we will spend more time analyzing and investigating ATP synthase and oxidative phosphorylation in more detail, as well as look at cases in which disruption of this process occurs either to disease or poisons. I will see you then.